Thank you, Dr. Xia. Thank you, Dr. Nguyen. So now I'd like to open up to the audience here for any questions around this exciting technology and its potential uses in the future for agriculture.
Okay, I can talk about it. The Stereo-seq technology offers a powerful spatial resolved single-cell transcriptomic strategy for systematic study of plant biology, and it will enable us to understand plants at an unprecedented resolution and the cell-to-cell communication and cell response to stress. However, there are still limitations and challenges for Stereo-seq, and several improvements are expected by future effects. First, the cost of the current technology is relatively high and need to come down further. And in fact, our colleagues are working on this. Second, pre-sectioning is challenging for many plants or tissues. The sectioning method needs to be optimized. Third, the coordinates of each cell are recorded, and more analysis tools for spatial-temporal omics should also be developed, such as tools for constructing the three-dimensional atlas of plant organs and tissues. For the application of Stereo-seq in food and agriculture, I think the answer is yes, absolutely. For example, we are construct the spatial-temporal atlas of rice seeds, which will tell us the characteristics of various cell types of seeds, such as endosperm cells and alluvial layer cells, and it will provide us with the possibility to improve the quality and taste of the rice. In addition, the STOmics of stress response researchers, it will provide us with genes to improve the stress tolerance ability of crops. So I think the Stereo-seq is a robust tools to improve the food and agriculture research. That's my opinion. Thanks.
Okay, thank you. So I'd like to ask a question for Dr. Nguyen as well, and of course the audience. Please feel free to ask any questions around the technology or for any of us as well. So Dr. Nguyen, thank you for that exciting presentation on aging clocks. I think in the mouse development example that I have presented, as well as some of the work that you're doing with aging clocks, it really shows kind of some of the different, interestingly, just happens to show the application of Stereo-seq for both development and aging. So there's this theme of like the concept of time and its importance in in really understanding lifespans and everything. What kinds of potential translational applications for your research for the aging clocks do you see might potentially be very relevant for the aims of food and agriculture for different governments and policymakers?
Yeah, so I think in aging, one of the consequences of the aging process is the metabolic processes that get dysregulated. And if we understand well how the body use the nutrition source in order to have a healthy aging lifestyle, at the molecular level, that I think would be very, very useful in terms of application. And as we see, the statistics should be like 2 billion people gonna be above 60 years old, and these 2 billion people are gonna consume a very different categories of nutrition. If we know which nutrition content is more suitable for aged people, yeah. So that is one example. Yeah.
Thank you, Dr. Nguyen. Very interesting. I think I think Dr. Nguyen certainly highlights some interesting perspectives there, which is that there's a lot of different angles through which you can see the applications of this technology, how it may be pertinent to a lot of considerations that are relevant to food and agriculture around the world. I think who needs what kind of nutrition, and how do we make better nutrition? How do we make more sustainable nutrition? How do we reduce waste and losses through disease? How do we make I think all these things are some of the applications that are truly enabled by fundamentally understanding all aspects of the biology behind these behind people across the lifespan, across animals and plants across the lifespan. Okay, I have a question for another question for Dr. Xia as well. So you kind of proactively answered a lot of things about how you see this technology is relevant to food and agriculture. I think what's I'm curious. So for the examples that you mentioned about ongoing research in terms of the life cycles of these different plants and everything, could you give us a little more color, and could you tell us a little bit more about what is the approach you're taking to understanding the life cycles of these plants, and what are some immediate translational applications that you hope to create out of the results that you gain from the life cycle atlas data sets that you generate? Hello, Dr. Xia? Dr. Xia?
Okay. I hear you. Can you repeat the question?
Yeah, so I'll repeat it a little slower. So you mentioned the ongoing research you are doing in terms of the life cycle atlases that you're exploring for some of these model plants. Can you tell us a little bit more about how you are designing these experiments? For example, how are you studying the life cycle with Stereo-seq, and for example, across how many time points, and then also what do you hope to do with the data sets that you generate in terms of translational applications?
Okay. Yes, we have designed the spatial-temporal omics atlas research of Arabidopsis and the rice. We hope this research data will become an encyclopedia of plant spatial omics research. That the plant researchers could download the data acquisition and analysis using our database that will be established in the future. We will focus on the developmental stages, such as the embryo development. We will try to explore the regulator mechanism of shoot apex and root apex development. That is to say how this apex formation and development. We will also care about the mechanism of cell sink conversion in the seed matrix. Because this is quite related to the quality and the taste of the rice or the other crops. We also will design some experiments to know how this the plant tissues or cells respond to the adversity, such as salt stress and the temporal stress and other stress environment. We want to know how this cell, what the cells are changed to response to tolerance this stress environment. This is part of the project, and much more aspects, and we welcome the other partners and institutions participating in our project and move forward the project much more in depth. Thanks.
Okay. Yes. So I think it's very it's very interesting what Dr. Xia just said, because when it talks to sustainability and food security, I think a lot of people realize the importance of for example, better breeding and creating crops that are more drought resistant or more nutritious. But I think it's a lot of it has been done so far with a lot of experimentation, trial and error. Whereas for the first time, I think what single cell, spatially resolved single cell omics, when applied to plant biology, especially from both especially also from a temporal aspect, is going to be able to enable all of us to rationally to unravel the precise mechanisms at the cellular and biomolecular level across space and time, so that we can actually rationally produce solutions to improve food production and food nutrition, and therefore ensuring food security for countries, and also better nutrition and health for people around the world. So I think that's just another interesting application that I'm sure we'll see more and more in the future. I would also like to spend the remaining time just addressing some of the questions that are kind of piling up in the chat box here. So I'll read them out loud, and I'll also respond one by one, or refer them to the appropriate person to respond. So first question: you are talking about the optimization of the sectioning. Are there any protocols, resources to help with this? So there's to answer this question, so for Stereo-seq, we do have a a database of different tissue types and from different organisms that we have experience with. And it, of course, this database is constantly growing according to, and, as global efforts accumulate more experience with this technology, as every tissue type and every organism is different. But it really depends on the experimental design. So on one hand, if we assume that average size cells 10 microns by 10 microns, but some cells might be bigger and some cells might be smaller. And then if you get too thin on the slices, it would be fragile and brittle and you easily break. But if you have too thick, you might have multiple cells stacked up, in which case it makes segmenting the cells going to be more difficult. So, I think that there's no specific, one size fits all answer. Part of it depends on experimental design, but we do have some accumulating experience with that. And I think that, we would be more than happy to share our experiences with all of you that might be interested in applying this technology to the different experimental directions that you're working on. Second question, are you considering to deposit the published Stereo-seq data sets in one common platform, which can be coupled to analysis tools and publicly available? So for this question, yes. So all the papers that we have published in the last several months using the Stereo-seq data, those data sets are all publicly available for download, as well as you will be able to access the toolkits and the analysis platforms that we've developed as well in order to further in order to play around first and then also get better inspiration for how you may want to design your own experiments. Next question, very nice technology. Is the technology set up for targeted gene panels as opposed to whole transcriptome analysis? Very good question. So, we do recognize that there's many applications, or there's many analysis needs that may not necessarily require a whole transcriptome every time. So sometimes you just want to use maybe whole transcriptome for discovery purposes, especially with unfamiliar tissues, but then you might want to really narrow it down to specific targeted genes for subsequent experiments. Currently, we do not have that available yet, but we're certainly, it's certainly technologically feasible. We've just been focusing mostly on the whole transcriptome initially. Right now, some of the additional applications, like I mentioned in my talk, we're also adding on being able to do proteomics with up to 20 plus DNA labeled antibodies at the same time to look at proteins and transcriptome on the same slide, as well as immunofluorescence too. But we certainly hear a lot of ask for the targeted gene panels. So right now you can use that in combination with some of the other existing technologies out there to do combined analysis. But we can we'll also have that available in the future too. Next question: how many mRNA transcripts in the smallest bin? So, well, bin is really just measured by the number of DNA nanoballs along each axis. So if you say the smallest bin, I suppose that would have to be bin 2, which is basically just 2 nanoballs by 2 nanoballs, being 4 nanoballs total, and the area in between, which is really 500 square nanometers. Regardless of what bin size it is, the number of mRNA transcripts is really dependent on the capture, on the specific experiment and the amount of transcripts that were captured. So it really varies. It's not a standard number, but for some of the for some of the really good data sets we've been able to generate, we've been able to see to see up to close to 2,000 transcripts for bin 10. So which is sorry, for bin 20s, which is about 10 microns by 10 microns. So if you talk about the smallest size, if you say bin 2, that's 1/100th of that. We've been able to get something like 20 or a couple dozen transcripts for the smallest bin in some of these data sets, but it really varies. Are there plans for implementing, for example, membrane staining or similar to optimize cell segmentation? This is another question that we hear very often, because similar to what you've been able to see that Dr. Xia had demonstrated with the Arabidopsis, people want to do the same with membrane staining especially for animal cells. We currently haven't. We don't have a solution for that yet, but certainly we are open to to people playing around with their own protocols we're co-developing, and of course we hope that in the future we'll also have something to better define the precise locations of the cell boundaries. So, okay, is this one of the pipelines, the GitHub BGI-Research Stereo-seq? Yes, that's the Python-based toolkit that we use to for the analysis of the Stereo-seq data. Can you also analyze methylation and other modification in plants? Dr. Xia, do you have a answer for this? Keke, can you also analyze methylation and other modifications in plants, other epigenetic modifications of plants with Stereo-seq?
Okay, it's the future aspects, but right now this technology is still ongoing. We couldn't do it right now, but we hope we can do it the next year or the future. And this question is quite important for many regulation mechanism is not just transcriptional, and other regulation ways is really need to be known. We are working on it, and we hope we can make it. Thanks.
Okay. And then next question is following on Ko's protocol question. Do you have optimized protocols for lung or liver cancer tissues? Lung and liver cancer, of course, they're very different tissues. Lung tissues have its challenges given the physical qualities of lung tissue, and for liver cancer, also different. We have some experience with liver cancer tissues right now, not that much with lung. So we wouldn't say that we have enough experience to have optimized protocols yet. Certainly many groups around the world have started working on these types of things. So you're welcome. So there's too many different combinations of tissues and organisms and applications and custom designs. So I think a lot of our support for this technology is really just in working together with you to find the best way forward, or to help optimize it. We have a team of field application scientists around the world that can assist with optimizing the protocols together with you. Then next question, thanks for the talks. Do you have a rough estimate of the cost of a 1 square centimeter? Is it compatible with Illumina NGS, or for obvious reasons, the readout was always DMDC? Okay, good question. So the cost for 1 square centimeter, if you were to buy the consumables yourself, is approximately $2,500 for just the chip alone. Of course, that does not include sequencing costs and everything. For the service costs, end to end, it's approximately $10,000 for 1 square centimeter. That means sample, tissue preparation, all the way to production of the results. These are just list prices. And then Is it compatible with the Lumina NGS? Currently, no. So this technology is only compatible with DNB-seq for now. But we have many solutions to make that available for you. We can take once you produce the stereo-seq, go through the stereo-seq library preparation step to produce the cDNAs. Once you could use our chips to do the tissue preparation from the tissue preparation to the cDNA step, and then you can either do the cDNA sequencing yourself, or you can send it out to some of the other parties around the world that would be able to do the DNB-seq for you. StereoPie BGI is an open access tool for, this is just a response from Dr. Xia. Okay. Okay. Yes, I see we don't have any further questions, and I see our time is just about up. Are there any last questions for me, for Dr. Nguyen, for Dr. Xia? Okay, so if there's no more questions, I think then we can end. Oh, freezing chips, is that possible? I'm sorry. What exactly do you mean is freezing chips? Oh, so I have to maybe there's confusion around the process. So I see freezing chips, collecting several chips over time and processed later. So the chips are not frozen. The chips are just come in like a box of the chips. They're silicon chips with the DNA nanoballs already sequenced and ready, and the probes attached and ready for capture. It's more about the collection of tissues. So, okay, yes, tissues. So tissues. We currently primarily work with fresh frozen tissues. So they're tissues that are collected fresh and embedded in at cold temperatures and embedded in OCT. We want to make sure that tissue preparation is very stringent, because it's important to maintain the RNA integrity. Without you minimize RNA degradation, or else you're not going to get much transcriptome information. So it's we've been able to work with fresh frozen, with flash frozen, and so you can certainly collect tissues first and then process them later. We would say don't slice the tissues until you actually need to use them. There's no need to slice them. Well, we can always slice them when it's when it's time to actually do the experiment. That's probably better. And then I also mentioned in one of my later slides that we will be also making this available for FFPE in the coming months as well. So that will dramatically increase the number of clinical samples that people have access to for research. Do you provide data for each chip? What locus specific barcode belongs to what position on the side? Yes, we do have. So each chip for Stereo-seq has a code on the back of the chip, and then that code, is the for that specific chip has its own specific file that that codes for the specific spatial locations of every single barcode. So that we call that a mask file. That's unique to every chip, and that's provided together with the chips that you use. Okay. Well, thank you very much. I think since we're already a little slightly past time right now, I think I think I'll hand this over to the organizer now. And I would like to thank our speakers today. Thank you, Dr. Nguyen, for making the time and for the great talk on the aging clocks. And thank you, Dr. Xia, for the presentation on the applications of stereo-seq in plants. And thank you to the United Nations FAO for the event and for the opportunity. And thank you to the audience as well for attending and for learning more about us. And we would be happy to talk to you more if you're interested in any further discussion. I will type my email into the chat box right now. And thank you very much.
Okay. Thank you all. I think that's the end. And if you have any further interest for STOmics, please follow us on the Twitter and the LinkedIn. The name is STOmics. Thank you. That's all.
Thank you all. Thank you.
Thank you. Bye.
Thank you, Kwan. All right.
Thank you. Have good
Bye.
sleep, Rabin. Thanks for staying up very late.
Yep. Signing off from California. Okay, bye.